So DNA stands for deoxyribonucleic acid, and right now we're going to talk about the nucleic acid part, right? Um, Nucleic acids are made up of three parts. We have the phosphate group right here, the pentose sugar right here, right? Pentose because you can remember it two ways. I remember it two ways. It has 5 carbons for penta, right? For pentose sugar. And also it's a 5-membered ring right here. See, 1, 2, 3, 4, 5. So pentose sugar. And then the part that makes everybody different is the nitrogenous base, right? That's where the genetic variation, the DNA sequence comes in to play. Whereas the phosphate group and the pentose sugar is the same throughout your DNA molecule. There are a couple different nitrogenous bases. The first one is a pyrimidine. And a pyrimidine is a single 6-membered ring with nitrogens in it. Single 6-membered ring with nitrogens in it. A purine, though, is 2 rings, right? One 5-member ring attached to a 6-membered ring with nitrogens in it. Okay? So when we're talking about the nitrogenous bases, we're talking about adenine, guanine, cytosine, and thymine, right? Turns out that adenine and guanine both are purines, right? Because they have double rings that look just like this, right? And then cytosine and thymine, right, are single rings. So what are they? They're the pyrimidines. So the easy way to remember which is that pyrimidines is the longer name and it goes with a smaller molecule. Purines is a shorter name and it goes with the bigger molecule, right? And then when RNA comes into the question, the only difference is that instead of having a thymine there, you have a uracil there, right? But uracil is still a pyrimidine. Okay. All right. So let's take a closer look at nitrogenous bases or DNA and RNA bases, right? And also the pentose sugars. So as we already discussed before, adenines, guanines, cytosines, and thymines are the basis of DNA. And then adenine, guanine, cytosine, and uracil instead of thymine are the bases of RNA. Down below we have the structure here for cytosine, uracil, and thymine. Notice they are all one single rings, right? And those are the pyrimidines. Remember that. Also remember that adenine and guanine are purines, and that's because they have 2 rings, right? I'm not going to make you memorize the structures of each one of the nitrogenous bases, but I do want you to know whether or not they are pyrimidines or purines. Then also, talking about the pentose sugar, right? Let's talk about the pentose sugar. Pentose because it has 5 carbons, 1, 2, 3, 4, 5, right? But the difference between RNA and DNA is the ribose versus deoxyribose, or ribonucleic acid or deoxyribonucleic acid. And that refers to what is on the second carbon of the sugar. So for DNA, there's a hydrogen there on the second carbon, okay? Whereas on RNA, there's a hydroxyl group. So that's got some very interesting implications, because you want the molecule that's holding all your information for life, right, which is DNA, to be as stable as possible. That's the reason why you have movies that kind of stretch the imagination and say that you can, extract the blood from a mosquito from 65 million years ago that was preserved in amber, and somehow you're able to pull out dinosaur DNA, right? But I guess more realistically, excuse me, we can actually take samples of DNA from items of evidence that are decades old and are still able to produce viable genetic profiles from that, right? And that's because DNA, by its nature, is very permanent or very stable, and that's because of this H. And I'll tell you why in just a second, because RNA is the exact opposite. Our RNA is actually very, not unstable, but very ephemeral or temporary in nature. And the reason why is because RNA is not used for the storage of genetic information, but it's used for the translation of that genetic information into amino acids, which eventually become your proteins, right? So when the DNA makes RNA, and the RNA makes the amino acids that become the proteins, your body needs to stop having that RNA around after the protein's already made. You don't need to have that RNA around anymore, right? Too much protein or enzymes of any single kind or anything that could be very detrimental to the health of the cell. And so that's why RNA has got to be able to be kind of like self-destructive, or, like I said, ephemeral or temporary. And the way it accomplishes that, the way nature accomplishes that, is by having RNA have this hydroxyl group on the second carbon of the ribose, right? Recall from the previous slide that the DNA backbone is made of a phosphate or a phosphodiester backbone, right? And then I'll just put R up here since I don't have any room. R, right? And this phosphodiester backbone, we call it diester because there's two esters right there, right? And then phospho because there's a phosphorus in the center, right? Like that. And then the phosphodiester backbone is attached to the DNA or the RNA like this, right? So you have the CH2 right there, and then we have the pentose sugar right there, and then the base. Okay? Same thing down here. Running out a little bit of room there, but that's okay. We only need one. And then the base, right? That's basically two nitrogenous bases, right, from a section of DNA. Okay. So on RNA, the second carbon has an OH, right? And the way DNA is twisted, which you can actually see back here on my Lego molecule. It's hard for me to point. Oh well, the scene just changed. But the way DNA is twisted in the double helix, right? It makes it so that this OH is in the perfect position to do a nucleophilic attack on that phosphorus right there, which is actually charged, by the way. It's got a plus sign on it. All right? And when it does that, it actually breaks the phosphodiester bond. But for DNA, which will make this one the DNA nitrogenous base, they're never on the same strand, by the way, so don't get confused there, but just say that this is DNA. There's no oxygen there to do that kind of attack on the phosphorus, right? And since there's no oxygen there, there's no real way to break that phosphodiester bond, right? So my old boss at school used to say, phosphorus loves oxygen, which is very true, right? Very true. And so that's why having this oxygen here makes it so that RNA can be ephemeral. Whereas DNA, having an H there instead, makes it much more permanent, right? Much more stable. So those are the implications, right? Long story short, or the thing you need to take away from this, is that ribose is temporary, ephemeral, whereas DNA, or deoxyribose, right, is much more permanent. Okay, much more permanent. So let's talk about nucleosides now, right? Basically, what I want you to know about nucleosides is that a nucleoside is the pentose sugar combined with the nitrogenous base. And they're combined together with a beta N glycosidic bond, right? And that's what I need you to remember about nucleosides. And the reason why is because there's also another thing called nucleotides, right? And the difference between a nucleoside and a nucleotide is that a nucleotide also has, in addition to the pentose and the nitrogenous base, has the phosphate group attached, right? Has the phosphate group attached. So things like this, which is adenosine monophosphate, right? Or it could be deoxyadenosine monophosphate, because if there's an H there instead of hydrogen, right, in RNA. Or guanosine monophosphate, right? Or cytidine monophosphate, uridine monophosphate, and deoxythymine monophosphate, right? So that's the thing I want you to remember, is that the difference between a nucleoside, right, nucleoside, and a nucleotide is that a nucleotide has the phosphate group attached, whereas the nucleoside does not. It's just the pentose sugar and the nitrogenous base, right? So just a quick summary about DNA and RNA. Bases are adenine, guanine, thymine, cytosine. Sorry, I said that backwards. That's cytosine, thymine. Or in RNA, guanine, I said it backwards again. Adenine, guanine, cytosine, and uracil. The sugar in DNA is a deoxyribose, which means that it's got an H there instead of an OH, right? At the bottom, 2 carbon, the 2 carbon of the pentose, right? But on RNA, which is ribose, that 2 carbon has an OH, right? That's the difference. Remember that. Remember that the nucleoside in DNA is the base plus the sugar. Same thing with RNA. It's the base plus the sugar. And then the difference between a nucleoside and a that's a typo. That should say nucleotide. Is that the nucleotide has a base, the sugar, and the phosphate attached, right? Same thing here, base, sugar, and phosphate attached. And then nucleic acids are a linear chain of nucleotides. So that's that strand of DNA I'm talking about, right? Same thing with the RNA. It's also a linear chain of ribose nucleotides, right? Same thing. Let's see here. Next is... So let's start naming some nucleosides and nucleotides, right? So DNA nucleosides add deoxy to the beginning of their names. And then RNA and DNA nucleotides are given by their 5' monophosphate, right? So real quickly, adenine, if it's a nucleoside form in DNA, is deoxyadenosine. In its nucleotide form, it is deoxyadenosine monophosphate. Guanine in DNA in nucleoside form is deoxyguanosine. In nucleotide is deoxyguanosine monophosphate. We use these abbreviations a lot too, the DAMP and the DGMP, also the DCMP, and then the DTMP, right? Moving on to the bases, right? Moving on to RNA. Adenine is just adenosine, whereas its nucleotide form is adenosine monophosphate, right? No D like there was here, okay? Cytosine here is cytidine, so CMP is cytidine monophosphate, right? So the names are pretty self-explanatory. Just remember that whenever it's a DNA molecule, in nucleotide form, there's always a D. RNA, there's not a D, right? And then the rest of it is just the initial of the base, like adenine, and then the MMP is the monophosphate part. Which is self-explanatory because there's only a single phosphate group attached, right? Single phosphate group attached. So let's talk about the nucleic acid structure. So nucleic acids, like I said, are the entire strand of DNA molecules, right? With the multiple pentose sugars in a polymer, okay, with its nitrogenous bases. And so polymers are many nucleotides in which the 3' hydroxyl group of the sugar in one nucleotide bonds to the phosphate group of 5' carbon atom in the sugar of the next, right? So this is actually the opposite of what I showed you before with the RNA breaking sugars or breaking the DNA, breaking the nucleic acid apart. We're actually bringing nucleic acids together now, right? So quite literally, you can have a nucleotide right here, right? And then in the presence of some acid, okay, the oxygen here can do a nucleophilic attack on that carbon, right? You end up making a water, right, from these guys right here, the acid and the hydroxyl that was on the 3' carbon, okay, and then you form an ester right here, the phosphodiester linkage, as I was saying before, right? So the important thing to remember about this is that the link between the sugars of adjacent nucleotides is called the phosphodiester bond, which I talked about before already. Remember that. So onto the DNA or the nucleic acid sequence, right? Nucleic acid sequence is read from the sugar with the free 5' phosphate to the free 3' hydroxy group, right? So this is the 5' end. Excuse me. This is the 5' end of the nucleic acid, and this is the 3' end of the nucleic acid. And the reason why it's like that is because the carbon on this side here is the 5' carbon, or it's the 5th carbon, right? 1, 2, 3, 4, 5. That's the furthest carbon, right? That's first. And on this end, the furthest carbon is the 3' or the 3rd carbon, right? And then DNA, the sequence of DNA, in this case adenine, cytosine, guanine, and uracil, is read from 5' to 3'. Okay? That's how it's read. Okay? And remember, we have our phosphodiesters linking each one of those nitrogenous bases or nucleotides. So a quick history about DNA, right? During the 1940s, scientists had already determined that DNA had the relationship of adenine to thymine and guanine to cytosine because they had equal amounts of each in a particular sequence, right? So in Watson and Crick, along with Roslin, by the way, they didn't even mention Roslin here, so I'm going to put Roslin in there. I don't think that's how you spell Roslin. That's embarrassing. Oh, my nose is running, so I need to stop that again. So a quick history about DNA. In the 1940s, scientists had already discovered that adenine and thymine went together in the double helix of DNA, and guanine and cytosine went together in the double helix of DNA, right? And the way they figured that out is when they determined the composition of DNA, they found that there was an equal amount of adenine to thymine and an equal amount of guanine to cytosine, right? So a really easy way to see the ratio, right? What goes to what. But then, in 1953, James Watson and Francis Crick, which they don't even mention Franklin Roslin here, but she was also instrumental in discovering the actual structure of the double helix of DNA, right? Don't forget Franklin Roslin. She was there. She was an X-ray crystallographer, and if it weren't for her work, Watson and Crick never would have figured it out. Very, very, very important scientist, Franklin Roslin is. She is very important. Anyway, the three of them together discovered that DNA was a double helix, right, that consisted of two polynucleotide strands that wound around in a spiral staircase, which is the double helix, right? This little guy right here. Let's see if I can draw it. I'm never a good drawer. That's close enough. That looks like DNA, right? It contained the sugar phosphate backbones that are analogous to the staircase rails, right? Rails on a staircase, with the bases arranged like steps along the inside, right? And they are actually anti-parallel to each other, right? Where the 5' to 3' end is bonded to the 3' to 5' end in that direction, right? So if we were to look at a DNA molecule, let's see if I can draw it again here. A double helix like this. Oh, that looks really good, actually. Right? On this end would be a 5' end, right? But over here would be the 3' end. Okay? And then this would be the 3' end down here for the first strand. And then this would be the 5' end down here for the second strand. And that makes your double helix, where the nitrogenous bases are hydrogen bonded to each other in between the double helix, right? And remember, DNA is read from 5' to 3' end, right? Remember that. That's important. And don't forget Rosalind either. So, like I was saying before, they complement each other. They go together, right? A's and T's go together. Adenines and thymines go together. And the guanines and cytosines bond together. How do they bond together? With hydrogen bonds, right? An adenine, it's a hydrogen bond from this amine group to this oxygen, this carbonyl. And also the hydrogen bonding between this nitrogen, this hydrogen on this secondary amine and this nitrogen, right? But the thing I want you to notice is that between the bond, the hydrogen, between the adenine and thymine are actually two hydrogen bonds, right? Two H bonds, right? Compare that to the bonds between cytosine and guanine. Very similar, right? Carbonyl hydrogen bonding to the amine right there. There's very similar hydrogen bonding happening there and happening there as well. But the thing that's different that I need you to know is that there are three H bonds here. Okay? Three hydrogen bonds. So you can then safely state that the bond, right, between the hydrogen bonding, or the bond between guanine and cytosine, is slightly stronger than that of the bond between adenine and thymine inside the double helix. And that has very interesting implications as well too, right? So we'll talk about this in more detail later. But there's this thing that we do called PCR, or polymerase chain reaction, okay? And it's when we take a targeted sequence of DNA from a larger sequence of DNA, right? A gene, right? And we amplify it from a single molecule of DNA to millions of copies, right? And we amplify one section of it. So one very specific that we are interested in section, right? So to do that, we actually have to do many analyses. And one of those analyses is called the GC content, or the guanine cytosine content. Because the more hydrogen bonds there are between the two nitrogenous bases, the harder it is for us to unzip it, right? Because you have to unzip the DNA before you can actually access the genetic information and see what the nitrogenous bases are. And so if there's a certain sequence that we're targeting in DNA, a strand of DNA, that we want to amplify, and the GC content is too high, it makes it very, very difficult for us to be able to amplify that, or even create primers to target that, right? And we'll talk about more about primers and amplifying and PCR later in the lecture. But that's the practical implication of having 3 bonds to 2 bonds, hydrogen bonds, right? GC content is very important in doing molecular biology work, right? GC content. And we'll talk more about that later on. But just remember, though, between adenine and thymine, right, 2 hydrogen bonds. Between guanine and cytosine, 3 hydrogen bonds. Oh, yeah, and also I forgot to say, too, excuse me, adenine is a purine, and it bonds with a pyrimidine, thymine, right? And guanine is a purine, and it bonds with the pyrimidine cytosine, right, in the double helix of DNA, right? So it's not going to be that like that thing where purines stick together and pyrimidines stick together. No, they actually are. It's more like an opposites attract type of thing, where a purine is with a pyrimidine, and a purine is also with a pyrimidine again, right? When it comes to adenine and thymine and guanine and cytosine, but it's always going to be A with T and G with C. Okay, so remember that as well. So let's talk about the double helix that I keep on mentioning, right? The double helix is 2 strands that wind together, right, of DNA in a double helix shape. And notice 5' end on that side, 3' end on that side, 5' end on this side, and 3' end on this side, right? And then we take a closer look at each of these nitrogenous bases, and you can see here, right, that adenine and thymine together there, holding together with 2 hydrogen bonds inside the double helix. And then here with guanine and cytosine right here, right, being held together with 3 hydrogen bonds. Right? Remember that. Remember that. Oh, and then also this purple one here, this purple strand that this is the sugar phosphate backbone. Or actually, I like to call it the phosphodiester backbone, right? You can call it either way. Phosphodiester backbone, or the sugar phosphate backbone. They both work for me, okay? Next. Oh, some practice. I didn't know that was going to come up. Erase. This is easy. Let's see what so if this is the 5' end right here, and this is the 3' end of the DNA strand, right? We want to see what the sequence is of the anti-strand, which is going to be 3' here to 5' here, right? The opposite strand, right? So this is a thing that we'd like to call anti-parallel, by the way. I forgot to say that. Anti-parallel. The 2 strands that are wound together in a double helix have their sequences anti-parallel to each other, right? Which means then this is going to be A to T, G, A, G, G, right? T, T, A, G, right? Good stuff, good stuff, okay? Next. Ooh, let's talk about some DNA replication now, right? So I was talking about PCR before, or polymerase chain reaction. Well, polymerase chain reaction is very much based on DNA replication, right? Except when you do DNA replication, you're replicating the entire DNA molecule inside the cell. Whereas in PCR, or polymerase chain reaction, you're only replicating or amplifying, in this case, a section of the DNA that you care about, a single gene, right? From the entire DNA molecule. And I was telling you before, for you as a scientist to do PCR, or for your cell as a cell to do DNA replication, you have to be able to access the nitrogenous bases that are in between the double helix, right? Because right now, or not right now, not right there. Let me erase that. Like up here, right, where the 2 strands of the double helix are still together, right? You can't access that genetic information. So how do you access it? Well, you have to actually split them, right? And you have to unwind them so that the nitrogenous bases, you have to unwind them through their hydrogen bonds, right? And you can unzip them so that you can expose those nitrogenous bases on each strand, right? Using an enzyme called helicase. Helicase goes through, right? Excuse me, and unwinds the DNA through its hydrogen bonding all the way up the middle and unzips it. Okay? And then after that, a DNA polymerase can come in, and it's much more complicated than this, but this is what I want you to know for the purposes of this exam. A DNA polymerase comes in, right? Which is exactly what it sounds like, polymerase, make into a polymer, an enzyme that makes into a polymer, right? Comes in and starts putting together free nucleotides, right? Based on the unzipped sequence over here, or unwound sequence over here, right? To make a new strand of DNA, or we call it a daughter strand of DNA, based on the original sequence. The same thing happens on both sides, right? But remember, though, you're reading DNA from 5' to 3', right? And so this is the 5' end over here of that, excuse me, that strand. So DNA replication occurs in this direction, right? Whereas down here, this is the 5' end down here. So DNA replication goes up the strand instead of down the strand, right? With the polymerase. Again, it's much more complicated than that, but for the purpose of this test, that's all I need you to know, right? Helicase comes in and unzips the DNA, right? Or unwinds it from its double helix shape to expose the nitrogenous bases so that a DNA polymerase can come in, right? And bring in free nucleotides right here, right? Free nucleotides. And start polymerizing them in the sequence of the original strand in the 5' to 3' manner, right? Direction. Okay? And that complementary, that's all based on complementary base pairing, of course, where A's go to T's and then G's go to C's. Right? So if the polymerase sees that there's a C there at the very beginning, like right there, it's going to bring over a G, right? And then it sees a G there, it's going to bring over a C, and so on. Right? And then polymerize them all together to a second strand, or a daughter strand, right? Coolio. So, there's also RNA, right? What you saw previously was just DNA replicating, right? Not RNA. And like we said before, RNA is the same thing as DNA, except instead of thymine, it has uracil. And instead of having a hydrogen on the number 2 carbon, the 2' carbon, it has a hydroxyl group, making it a lot more ephemeral. Okay. Excuse me. And the other main thing that's different too is RNA is usually single-stranded. Okay? Compare that to DNA, which is double-stranded, and that's why it's a double helix, right? And then RNA molecules are much smaller than DNA molecules, and the reason why for that is because, like I was saying before, DNA will only become RNA, excuse me, will only code for RNA that's for a specific gene that makes a specific protein. And once you're done with that, when you're done making the protein you need to make or the enzyme you need to make for a particular function, you don't need any more of it, that RNA breaks down, right? Due to the fact that it has that that nucleophilic oxygen right there, right? Or sorry, electrophilic oxygen there, right? That wants to attack the phosphate, because remember, phosphate loves oxygen, right? So they're smaller. There are 3 types of RNA that you need to know about. There's mRNA, right, which is messenger RNA. It carries genetic information from DNA to the nucleus of the ribosome. Sorry, excuse me. It carries genetic information from DNA in the nucleus, right, to the ribosome in the cytoplasm. The ribosome is where amino acid synthesis occurs, or protein synthesis occurs, right? Or peptide synthesis, right? Then there's transfer RNA, right? Transfer RNA intercepts genetic information in the mRNA, right? And brings in the specific amino acids for the protein synthesis, right? Sorry, I mixed up there. There is something separate. Amino acid sequence. Sorry, amino acid synthesis is completely different from protein synthesis, right? Because when you bring in the amino acids into the ribosome, this is getting a little messy, so I'm clear this, okay? When you bring in the amino acids into the ribosome, so they can be attached to each other based on the RNA sequence, the transfer RNA sequence, those amino acids are already synthesized. They're already in the 20 different structures that the amino acids that you learned about last lecture about, right? They're already together and they're ready to be put into a peptide. There's also amino acid synthesis, right? Where you actually have to make those amino acids, and that's a completely different lecture, right? But here we're talking about protein synthesis from already synthesized amino acids, okay? And then we have the third type of RNA, which is ribosomal RNA. And this is the most abundant type of RNA. And the reason why is because it combines, these structures combine into secondary structures with other proteins to make the ribosome, right? To make the ribosome. So previously we didn't know that that RNA could actually become a secondary structure that's functional, right? But it turns out that it can, much like amino acids can, right? In this case, it's the ribosome that actually makes the protein synthesis, that carries out protein synthesis. So, quick little thing here, little review summary of RNA. Ribosomal RNA or rRNA is about 80% of all RNA in a person. And it is the major component of ribosomes in the site of protein synthesis. Messenger RNA or mRNA is only about 5%, which makes sense, right? The only purpose you have of messenger RNA is to take the DNA, is to take the information from the DNA in the form of RNA and take it from the nucleus where the DNA was and move it to the ribosome, right? So it carries information for protein synthesis from DNA to the ribosome, right? And then transfer RNA is what actually, it's 15% or tRNA, 50% in people. And it actually transfers amino acids, right, to the ribosome to make protein synthesis based on the, to carry out protein synthesis based on the sequence that's on the mRNA, right? So a quick thing about tRNA structures or transfer RNA structures, right? They're about 70 to 90 nucleotides long, and at the 3' end of the sequence, right, there's actually is where the amino acid is attached. And we'll talk more about that in a second, or in a few lectures, but that's where the amino acid is attached. One of the 20 that you learned in a previous lecture. And then the other important part of this tRNA structure is that right down here, the end of this, we call these like keyhole structures, right? Because they look like old school keyholes, or hairpins, or hair loops. You know, those kinds of things. We're very descriptive in biochemistry, obviously. Anyway, there are 3 nucleotides right here which represent the anticodon, right? So remember, I don't know, I can't remember if I spoke about this before, but every 3 nucleotides, and we'll see this in much more detail in a second, but every 3 nucleotides constitutes a codon, right? Which then translates to an anticodon, 3 more nucleotides that are their complement. And then that anticodon is attached to this piece of tRNA right here, right? And then that piece of tRNA is attached to an amino acid, right? So that's what you need to remember there. So anticodon is on the tRNA molecule. Okay, we'll talk more about this in more detail in a later slide, but I just wanted you to know just ahead of time that the amino acid's connected to the tRNA, and then you have the anticodon right there. So let's talk about protein sequence. Not sequencing. That's a completely different thing. protein synthesis, okay? Protein synthesis involves transcription, and what that is, it's the transcription of the DNA genetic material or nucleotides into a piece of RNA, mRNA, right? That's the transcription process. So the sequence that's in the DNA, like the A, T, G, C, T, T, A, that sequence, right, gets transcribed into a strand of RNA. Very similar, in a very similar fashion, that DNA is being replicated, except instead of using DNA polymerase, it uses RNA polymerase. Okay. So once transcription happens, and you get that DNA molecule making a mRNA molecule of the complementary sequence, you get a piece of RNA right here, right? Short, compared to the really long, DNA, right? After that, there is a process called translation, where the mRNA, right, leaves the nucleus and goes into the cytosol, or cytoplasm of a cell, and meets up with a piece of tRNA, or meets up with some tRNA, that has amino acids attached to it, right, in a thing called a ribosome. That's not how you spell ribosome. That's how you spell ribosome. Yeah, there we go. Ribosome. In a ribosome. And that's where translation occurs. Where the DNA sequence that got turned into an RNA sequence then becomes that same DNA sequence again in translation, but except since it's a piece of RNA, it's uracil instead of thymine, right? And based on that previous slide I told you, right, each 3 of those nucleotides here, right, are representative of an amino acid, because the tRNA has the anticodons attached to the amino acid, right? I know it sounds complicated. We'll get more detailed in a second, or not more detailed, but we'll look at it more deeply. So the genetic information in DNA is replicated in cell division, and is used to produce RNA that is the code for amino acids needed for protein synthesis, right? So those are the 2 processes that you need to remember right here: transcription and translation. Okay, those are the 2 processes. Transcription is DNA to RNA. Okay. And then translation is RNA to protein. Okay. Watson and Crick came up with this thing called the central dogma, right? And it's basically a triangle where DNA is at the top of the triangle, and then RNA is over here, right? And then protein / amino acids over here, right? So DNA can become RNA based on transcription. Transcription, right? And then RNA can become protein because of translation. Okay. And then DNA can make more DNA through replication. Right? Replication. There we go. Replication. Okay? And that's a central dogma. We have yet to be able to go from protein back to DNA. I mean, there's viral ways, like with reverse transcriptase and such. But naturally, I mean, I guess reverse transcriptase is a viral virus, and that's natural. But basically, it's DNA to RNA to protein, right? And you can go from an amino acid sequence in a protein using reverse transcriptase. It's an enzyme. That's getting very messy. Reverse. It's always like towards the end of the lecture where my Apple Pencil starts to die. Transcription. And that's done by an enzyme called reverse transcriptase. Okay? Moving on, sorry, went on a tangent there. Excuse me. All right, so let's go dive deeper here just to make things a little bit more complicated. No, I'm just kidding. Make things a little bit more simply. In the transcription process, right, a section of DNA containing a gene unwinds, and then an RNA polymerase enzyme uses one of the strands of the DNA to synthesize mRNA, right? So here in this picture here, we have the double helix, right? And it unzips right here where this gene is, okay? And RNA polymerase comes in, starts to read the sequence on the DNA, and after it reads it, it starts to synthesize RNA by reading the sequence and finding its complementary RNA bases, right? Good stuff. And that's mRNA that it's making. And like I was saying before, it uses the complementary base pairing, right, where it sees a G. Sorry, yeah, it sees a G there, so it's going to bring in a C. Oh, I got a little backwards here. It sees an A there, right, in the DNA sequence, and it brings in a U. It sees a T here in the DNA sequence. It brings in an A. It sees a G, brings in a C, sees a C, brings in a G, sees a T, brings in an A, sees a U, sees an A and brings in a U, right, instead of thymine, and then sees a G, brings in a C, sees another G, brings in this C, and then sees a C and brings in that G, right? And it keeps on going until it's done. That's done by RNA polymerase, okay? And once it's finished, there's a termination point in the DNA sequence. We'll see that in a second, right? And the mRNA is released. So let's talk a little bit more deeply now about the genetic code and how we make proteins using amino acids, right? So the genetic code consists of a series of 3 nucleotides, right, or triplets, also known as codons, right? In the mRNA, yeah, it says it right there, that specify the amino acid and their sequence in the protein. Codons have been determined for all 20 amino acids, the all 20 that you learned in the previous lecture, right? Needed to build a protein, right? And so at the beginning of the mRNA, the codon AUG signals the start, right, of protein synthesis, right? And then to stop, there's UGA, UAA, UAG, and that signals termination of protein synthesis, right? My favorite is UGA, because whenever you're actually coding it in DNA, it's TGA, right? I don't know why that's my favorite. That's a really weird thing to say. Really weird, right? Anyway, but yes, that's how you know. And so, just for an example, if we have these codons in the mRNA, UUU, right? That codes for a phenylalanine. More U's, another phenylalanine. 3 more U's, that's another phenylalanine, right? And there's a whole chart that shows you what codon goes to what amino acids. Oh, and there it is. How exciting, right? If you remember, let's see here. Let's see if we can find the start codon right here. There's the start codon right there. Remember I said that a long time ago, you get extra points if you remember this. A long time ago I said a methionine is a start codon, right? Well, there it is. AUG, the start codon, actually codes for methionine, right? So it's easy to read this chart, right? So, for example, if you're looking for the code, let's say you're looking for the code cytosine, adenine, cytosine, right? So what is that? What amino acid is that code for? So the first thing you do is I find the C, which is here, right? Then the second letter is here, A, right? So now I've gone over here and then go down here, right? And then the third letter, which is C, right there. And so the codon CAC codes for the amino acid histidine, right? So that's how you use this chart. And so every combination of these codons codes for the 20 amino acids that you learned in the previous lecture, right? And multiple codons can code for the same amino acid. This is a very handy chart for when you're trying to make your own proteins and do a lot of protein synthesis and enzyme work like I do. It's a very handy chart. Oh, and then also I forgot the stop codons. So we had our start codon here, which is methionine, and then TGA or UGA. So we'll start here, U, right? G, and then A, right there. Stop codon. See? So those are things I want you to remember. The start codon methionine, the stop codon UGA, right? And just basically how to read this chart. Okay? Oh, look at that. It's going to make us practice. Okay, so we have CCU. So C, let me clear this now since we need to see this chart completely. We have C, right, is the first letter. And then the second letter is C, which is this block right here, right? And then we don't even need to know what the U is because all four of these are proline. But just in case you wanted to know, there's U right there, CCU, which is proline. That's the first amino acid in this amino acid sequence, codon sequence. Then we have AGC, A right here, and then G, and then again, all four of these are threonine, right? I'm just going to write the three-letter thing so it's not so crowded here, right? But if you wanted to go further, C is right here, right? So ACC. Oh, I messed up. I thought it was AC. It's good thing I'm not in charge of protein synthesis. It's not AC, it's AG. So A, and then G, right? Oh, this one you have to go to the third one. So A here, and then G is here, right? G is here, so it's this block, right? And then C, which is right there. So that's serine, actually, not threonine. Interestingly, both of those are the hydroxyl on the alkyl R groups. So it'd be a minor amino acid change, maybe not too bad of a mutation. You know, maybe hopefully it's not a silent mutation because it's definitely a different amino acid. But activity might be comparable, maybe a little less on the threonine, right? But serine is actually very important in in catalysis. So enzyme catalysis. So that would be important to have the actual serine there and not mess up by saying that there's a threonine in it, right? Okay, GGA. So G here, and then G there. So that's this block, right? All four are glycine. So, but still though, A there. So that's glycine. And then CUU, C to U, this block right here. And all four are leucine, but CUU is down right there, right? And that's leucine. So using that chart, you know, just be able to translate from a codon or a DNA sequence to a RNA sequence to a amino acid to make yourself a little peptide chain right there, right? Activation of tRNA. I said we'd get close. So more on tRNA. I told you we'd come back to this, right? We need to activate tRNA for it to be useful in protein synthesis, right? And tRNA by itself, generally doesn't have an amino acid attached to it. But if you remember from the previous couple slides ago, I was saying that there was a 3' end of the RNA that is where the amino acid gets attached. Well, this is how it does it. There is an enzyme called tRNA synthetase, right? There's one for every amino acid. So this one specifically is called tryptophanyl tRNA synthetase. And it, along with some ATP, can attach tryptophan here, the amino acid, right, the one that makes you sleepy after eating a lot of turkey, to the tRNA at the 3' end, right, at the 3' end, using a very high energy bond, right? High energy because high energy, it's high energy, and that's why you need to use ATP to do it, right? But high energy because you need that energy, you need that bond to be able to be broken easily, so that you can attach this RNA, detach this tRNA, right? So that you can attach this amino acid to another amino acid in protein synthesis, right? So once this amino acid is attached to this piece of tRNA using the high energy bond, right, it gets floated over to the ribosome where it matches up to the mRNA with its complementary base pair, right? And that's how an amino acid, the next amino acid in the chain, gets attached to the ever-growing peptide chain that will eventually become a protein, right? So the thing you need to remember about this is that the enzyme is tRNA synthetase, right? It's tRNA synthetase. So here we have not a piece, but we have one tRNA, and attached to it is a serine, right, at the 3' end, like I was saying before, right? And again, initiation gets started, right, when you have a codon in the mRNA that's AUG, and the thing that the tRNA is brought that's brought over by tRNA is a methionine tRNA, right, and that starts the process of protein synthesis, right, or peptide synthesis. I use that interchangeably, remember? And then after that, you can elongate the chain from the methionine, say with a serine, right, by bringing in more tRNAs with the next codon, next complementary codon, right? And a peptide bond forms between the C-terminal of the methionine and the N-terminal of the second amino acid, right? So you use that high energy bond from the tRNA, from the amino acid connected to tRNA, right, to then form the peptide bond that holds two amino acids together, right, which was an amide bond too. Remember, it's this little guy right here from the previous slide. Right? So the initial tRNA detaches from the ribosome, which shifts to the next available codon, and that process is called translocation, right? And so during chain elongation, ribosomes move along the mRNA to form from codon to codon, so that the tRNAs can attach new amino acids to the growing protein chain, or the polypeptide chain, right? Just basically what I said, but in words for you there. Help you out a little bit, right? So the thing I want you to remember, though, is that the tRNA brings over the next amino acid that's in line, right, based on the codon that's in the mRNA, and it attaches the next amino acid to the original amino acid that started, right, that's before it, by using the amide bond, which is the peptide bond, right? It breaks off from the tRNA, like for example the serine, right, and attaches that serine to the methionine. Say that was the first one. It's always the first one, right? By using a peptide bond. That's what I want you to remember from this. Okay. So a little bit more, a little bit, just a quick thing about more about translation, right? So translation is the process of reading the mRNA and translating that into amino acid to anticodon, which then goes into amino acids, right? So in the process of translation, mRNA that was synthesized by transcription attaches to a ribosome, and the tRNA picks up their amino acids bind them to the appropriate codon and then place them in the growing protein chain, right? So if you look really closely at this picture here, we have a string of mRNA, right? That long strand of mRNA. It came from the nucleus right after it did some transcription from the DNA, right? And a ribosome attached to it. Right? Or it attached itself to a ribosome, right? And then the ribosome is responsible for bringing over the tRNA that has the anticodons for the mRNA's codons, right? With its corresponding amino acids, right? Its corresponding amino acids. Make sense? Good stuff. So how do we stop protein synthesis, right? How do we stop protein synthesis? Well, there's a stop codon, UGA, right? Or UAA or UAG. And what happens is that once it reads all these codons and reaches to the stop codon, right? A release factor recognizes a stop codon and makes the entire translational complex dissociate, right? So the ribosome and the mRNA all fall off, right? Once it reaches the stop codon, right? So, quick summary then of protein sequencing. I keep on saying sequencing. Protein synthesis, right? First, we begin with DNA transcription. That's where the DNA template is used to produce mRNA, right? That happens in the nucleus with nucleotides, RNA, and polymerase. Then you have to have the activation of tRNA, right? That happens in the cytosol or cytoplasm, where the amino acids and the tRNAs get bound together with the amino acyl tRNA synthetase, right? Molecules of tRNA pick up specific amino acids according to their anticodons. Translation of the mRNA is the third step, right? And that happens in the ribosome with the mRNA. mRNA binds to the ribosome where the translation begins, right? The codons get translated to anticodons. And then initiation begins. That's with the tRNA methionine, of course. If it's the first one it is the first one. I keep on saying if it is. It is the first one in the chain. And we have some mRNA there, of course, and some more amino acyl tRNA synthetases, right? Or tRNAs, because they've already been made by the synthetase in one of the previous steps. The start codon binds the first tRNA carrying the amino acid methionine to the mRNA, and then successive tRNA binds to and attach from the ribosome as they add an amino acid to the polypeptide, right, during the protein sequences. And then chain termination occurs with a stop codon that's on the mRNA. That happens in the ribosome. And so the protein is then released from the ribosome where it can go through and form its the peptide chain is released from the enzyme from the ribosome, where it can start to form its non-covalent interactions and covalent interactions to form the three-dimensional tertiary structure, secondary structures and tertiary structures, and even quaternary structures that become a functional protein or enzyme. Okay, so let's do a quick practice problem. We have a section of DNA, and we're going to use it to build some mRNA for a particular protein. And our sequence is GAA-CCC TTT, right? We want to transcribe that into mRNA. So what's our sequence going to be? It's going to be CGAA right? Because remember, this is actually mRNA, not DNA, so all the T's become U's. And then 3 C's in a row means there's 3 G's in a row, right? And then 3 T's in a row means 3 A's in a row, right? Make sense? So what are the anticodons for the tRNAs then, right? We got to translate this now to the anticodons, right? For CUU, it becomes back to GAA, right? And then GGG becomes CCC, right? And then AAA becomes triple U, right? Not T's, because this is RNA. And if we look up in our our chart, we look up in our amino acid chart which ones they are, right? Remember that in our amino acid chart, they're not anticodons that are in that chart, they're actually codons, so we need to go back to the codon, which are those. And so CUU in the codon chart actually is leucine. And then GGG in the codon chart is glycine. And then AAA or AAA translates into lysine. Not leucine, but lysine. So that's one of the mistakes people make. People try to look up the anticodon in the codon chart, right? Remember, this is the anticodon. You're going to get completely different amino acids when you look up those letters in the codon chart. So you need this one. You need the codon chart. Right? You need the codon chart. You have a codon chart. You need to use the codon, right? Yes. Right? So some practical stuff with DNA now. Turns out that there's a lot of antibiotics that inhibit protein synthesis. For example, chloramphenicol, right? This inhibits the peptide bond formation, preventing the binding of tRNA. There's erythromycin, right? This inhibits peptide chain growth by preventing the translocation of the ribosome along the mRNA. So as the mRNA is moving along, reading the mRNA, as the ribosome is moving along, reading the mRNA, right? That's called translocation, and this prevents translocation of the ribosome. Puromycin causes the release of an incomplete protein by ending the growth of the polypeptide early. There's streptomycin, prevents proper attachment of the initial tRNA. And then there's tetracycline, which presents the binding of tRNA at all. Now these are different antibiotics than what we talked about before, which are the beta-lactam antibiotics, right? Beta-lactam, right? And those were the antibiotics that had the 5-membered ring that was kind of unstable, or 4-membered ring that was kind of unstable, and it would actually prevent the formation of the bacterial cell wall, right? The peptidoglycan cell wall. So that's the difference between these DNA-based antibiotics and then the beta-lactam antibiotics like penicillin or amoxicillin, right? Things that you get like in for basic bacterial infections, right? There's another DNA one here called Flagyl, right? That's another really, I think that's how you spell Flagyl. I can't remember how to spell Flagyl, but that's another very powerful DNA antibiotic. So just remember the difference, right? The beta-lactams, or the penicillin-based ones, right? They act on the cell wall, preventing the cell wall, peptidoglycan cell wall of bacteria from growing. Whereas these DNA ones actually prevent DNA synthesis. I'm not sorry. Protein. Let me say that again. These protein synthesis ones actually prevent the the proper formation of a peptide chain to turn into a protein, right? And then there's flagell, which actually inhibits DNA synthesis. Dangerous stuff, right? Mutations, right? A couple of things can cause mutations in DNA, which are not good, right? Because you mess up your DNA sequence with a mutation, and you can get a completely different amino acid in there, or lose one, and that's very bad, right? So things that can cause DNA mutations, X-rays can do it, and whoops, I forgot to change it to pencil mode. So let me try that again. 1 second here. There we go. Okay. So things that can cause mutations, right? X-rays can definitely cause mutations. That's why you got to wear a lead vest whenever you get an X-ray, right? Also, ultraviolet and UV light can, they're the same thing, ultraviolet and UV. It's not or, they are the same thing. Can cause mutations as well. And if you remember what I was talking about, man, what lecture was that where I was talking about the different types of electromagnetic radiation, right? And the energy that is in UV light, right, or ultraviolet light, is it corresponds to the energy that holds together a carbon-carbon bond, right? And for X-rays, that energy exceeds the energy that's in a carbon-carbon bond, right? And so that's why things like that, X-rays, and even gamma rays, right? Because you can get the Hulk from gamma rays. Man, his DNA got mutated. And then UV light, right? All those are either the energy associated with those types of energy are either at the same level as a carbon-carbon holding together a carbon-carbon bond, right? Or they exceed that. And that's why these types of electromagnetic radiation can cause mutations in your DNA. Because you remember the structure of your DNA, most of it, right, is what? Carbon. Except for the phosphodiester bond and the nitrogenous base, the backbone is a sugar. 5 carbons, right? So that's why there. Viruses can do it too. Chemical mutagens can do it too. There was this one molecule or what is it called? Again, I forget what it's called now. Acrylamide. That's what it's called. Acrylamide was a thing that we used to make gels to do electrophoresis. So polyacrylamide gel electrophoresis. And the polyacrylamide, which is actually a solid piece of gel, wasn't quite dangerous, but the bis form or the monomer form, right, was super mutagenic, right? You could get cancer from touching it with your skin. And there was this one guy in my lab when I was a graduate student. He was making some polymerized, acrylamide for the page, the polyacrylamide electrophoresis, right? He was making some from the monomer, which is just acrylamide, or also known as bisacrylamide, and he dropped a flask, and he starts to clean it up, but like there's broken glass everywhere on the table and on the floor, and he starts to feel around for it using his glove, but instead of picking up the glass, like a normal person would do, him and his gloved hand was like patting down and then streaking his hand across the floor to feel for the glass. And I'm like, dude, what are you doing? Like, if you cut your glove open, you're going to expose yourself to the acrylamide. You're going to get us all killed. Well, not all of us, but him. Turn us all into mutants, which isn't a bad idea. I wish I was a mutant, kind of. Maybe a Professor Xavier type of mutant. I digress, sorry. Anyway, the point is, very dangerous. Bisacrylamide, very dangerous. The polymerized form, not so dangerous. But either way, you want to be very careful with that, right? So if a mutation occurs in a somatic cell, a cell other than a reproductive cell, the DNA is altered, will be limited to that cell and its daughter cells, right? So the good thing is that you have a gazillion cells in your body, right? And if only some of them are exposed, right, then only those ones will have the altered DNA, and their daughter cells after mitosis will have the alteration too. But hopefully the vast majority of your cells are still normal, right? So mutations cause uncontrolled growth, and cancer could result, right? If a mutation occurs in a germ cell, So that's the difference, right? We have a somatic cell here, which is anything but a sex cell, and then the germ cell, which is a sex cell, egg or sperm. Then all of the DNA produced from that new individual, will contain the same genetic change. Yeah, so be careful with that. This is, this is a little picture of a, mutation there. I don't know what mutation, but it's there. So there's a few types of DNA mutation. We have the point mutation, right? We have the silent mutation. We have the deletion mutation, and we have the insertion mutation, right? A point mutation is a replacement of one base in the template of the DNA strand with another, right? So, say it was supposed to be a thymine, but it got replaced with a guanine, and then you have a point mutation. We actually do that a lot in molecular biology because we want to be able to do like activity assays on different proteins and figure out which amino acids are vital for the function of an enzyme. So what we'll do is we'll figure out what the sequence is we'll figure out what the DNA sequence is of a particular enzyme or protein, and then we'll zero in onto the section of that DNA sequence that's the active site, and we'll start changing those what we call active site amino acids, what we think are like really what's involved in the reaction, and we'll change them to different amino acids. So say there's a serine that's in the active site, and we think that's a catalytically active amino acid in the active site of an enzyme, right? Instrumental in causing some kind of enzymatic reaction to occur in a cell. So what we'll do is we'll change the sequence. We'll do a point mutation called site-directed immunogenesis, where we'll change the codon, right, that codes for that serine that we think is important, and we'll change it to the codon for like something like glycine, right? And that way, the OH is gone from the serine, which is we think is catalytically important. The only thing there now is a hydrogen, right? And we'll see if the enzyme is still active. Can it still catalyze the reaction? And if it can't, then that means that serine, that OH, was important, right? And so that's how we tell what enzymes are important and what enzymes are not important. Sorry, that's how we tell what amino acids are important and what aren't important inside of an active site. And in the previous lecture, I was talking about how the difference between having an OH and an H makes DNA more stable, vastly more stable than RNA, right? So that change from an OH to just an H can really let us glean some insight into the active site of an active site of an enzyme, right? And so that's what a point mutation is, where you change one base and you get a different amino acid. And this can happen naturally, but we can also do it like in the lab as well. Then we have a silent mutation. A silent mutation is basically a point mutation that doesn't change the amino acid. Because remember, in the amino acid chart, the codon chart, multiple codons can make the same amino acid. And if you just happen to change do a point mutation on one of the bases and it doesn't change the amino acid, that is known as a silent mutation, right? Deletion mutations are bad because that means that one of the bases are completely gone. It shifts the entire, what we call reading frame, over, and now all the amino acids after that deletion are different. That's like catastrophic for an enzyme. And then the same thing can happen with an insertion mutation, where instead of having a deletion, you have an extra added base, and then everything after that is frame shifted, and it's catastrophic. And the picture I have here is UV coming in and attacking. I have no idea what these colors mean, right? But they're AGs, T's, and C's, right? But the UV comes in, and this is called actually a DNA lesion, right? Where two bases that are on the same strand attach to each other. And that happens from DNA. That's a result of RNA. No, that's a result of UV light, right? And this kind of thing can cause cancer, right? That's why you should stay out of the sun too much. So let's go through and look at these just, very specifically, and more closely. So here we have the point mutation, right? And in the point mutation, you notice right here, right, We have our stop codon, okay, like we were talking about in the last lecture. We have our start codon in the normal DNA strand, right? The start codon methionine, right? But here in the normal DNA strand, we have a glycine, right? And the code should be CCG, translating to GGC. But since it's a point mutation, one of the C's got changed to a T, and now the anticodon is an A, and so instead of getting a glycine there, you get a serine, which is very similar to what I said before, changing a serine to a glycine, right? But this one is changing a glycine to a serine, right? And that's called a point mutation, right? You see how changing just one amino acid, one nitrogenous base or nucleotide in the with using a point mutation, and you get a completely different amino acid, right? So that's definitely not silent. And there's no telling what this can do. But I will say that glycine is very different from serine, right? Serine is a polar amino acid, and glycine is a nonpolar amino acid. It's as different as it can get, right? Bless you. That was my dog. He just sneezed, or she just sneezed. I have multiple dogs in the room right now, 3 total, 2 boys and 1 girl. And the girl, her name is Kiara, and she's like rolling around on the couch right now and sneezing. So excuse her. Bless you. Anyway, so the change of one nucleotide here to a T changes this to serine, and serine is polar and glycine is not polar, and that's as different as you can get, right? And so this right here could be, I don't know if that could be an active site kind of change where it's no longer active, but remember that glycine has just got the H there as its R group, and then serine has got the OH, right? And the H makes it where glycine is small, and in a long chain, when there's a bunch of glycines, you can get them to kind of stack on each other and make some secondary structures. But if you have an OH there, which is bigger, it might try to, the secondary structure might not form correctly anymore, and then you have an inactive enzyme, right? And that's just me speculating, especially since it's only like 5 amino acids long. So, next, we have a deletion mutation, right? So again, we have our normal DNA strand here, and this time, right here, we have AAA as the codon. The anticodon becomes triple U, coding for a phenylalanine, right? But in our deletion, one of the A's is gone, right? And like I was saying before, what happens there is that everything after changes, because the entire reading frame got shifted to the left, okay? The codon frames got shifted to the left. And so now, instead of having a phenylalanine there, you have a leucine there, right? Which both are nonpolar, but leucine is an alkyl, and this one, phenylalanine, is a benzene ring, right? So those are very large, though they're both property-wise the same because they're nonpolar, they're very different in size. So that could cause some structural issues with the secondary structures or tertiary structures of an enzyme or protein. Everything else stays the same, though, right? So the other thing I forgot to mention too is that since we have a complete frame shift over to the left, and everything behind the mutation has changed, amino acids, we no longer have a stop codon there anymore. So this thing could just conceivably keep on synthesizing a peptide until it finds another stop codon, and you really got some problems. So deletions are very bad. They're definitely not silent. Then we have an insertion, same thing, right? Except instead of it being a frame shift to the left, it's a frame shift to the right, right? Here, we lost the T, or sorry, we didn't lose a T, we inserted a T, and when we inserted a T, we split up the AAA that was supposed to be making phenylalanine, and now we've got leucine and tryptophan out of nowhere, and another leucine. No more glycine and stop codon. So when is this guy going to stop? Right? So that's an insertion. That is an insertion mutation. Those are also very bad too, and they definitely are not silent because you don't know when there's going to be a stop codon to stop it. And even though these are like leucine is still these are nonpolar and stuff, right? Tryptophan is nonpolar as well. Even though they're nonpolar, they're they vary vastly in their shapes, right, in their sizes. So that's a problem. Bad insertion mutation, very bad. So, what are some of the effects of mutations, right? I alluded to most of this already in the previous slides, but a change in the amino acid sequence can change the structure of the protein, severely altering it, and causes loss of biological activity, right? When this condition is hereditary, it's called a genetic disease, but the single mutation from X-rays, mutagens, UV light, and viruses can cause the alteration of DNA, making a defective protein, and you'll get cancer if it's affecting somatic cells, right? And it becomes a genetic disease if it affects germ cells or sex cells, right? Eggs or sperm. So, keep your germ cells or your sex cells safe from, UV light and X-rays, right? Keep those safe, because then your offspring might have mutations. A genetic disease is a result of a defective enzyme caused by a mutation in its genetic code. So, for example, you can have phenylketonuria, which results when the DNA cannot direct the synthesis of an enzyme, and the enzyme phenylalanine hydroxylase, required for the conversion of phenylalanine to tyrosine. Ooh, that's bad. So phenylalanine, remember, is the R group that looks like this. My favorite amino acid, right? And then tyrosine is the R group that looks like this. So that's important. Why? Because that OH group there makes tyrosine polar, right? Whereas phenylalanine is nonpolar. Albinism, like albino, albinism results when an enzyme that converts tyrosine to melanin is defective. Oh, that's not good either. Right? Let's keep on trucking. Let's take a look at some genetic diseases, right? We've got galactosemia, right? And this is when a transferase enzyme that is required for the metabolism of glucose 1-phosphate is absent, right? Resulting in the accumulation of galactose 1-phosphate, where it leads to cataracts and mental retardation. That's bad, right? It's seen in about 1 in every 50,000 births. We've got cystic fibrosis. I actually did a lot of research on cystic fibrosis when I was in grad school. And this disease is when you're missing an amino acid in your lungs, right? And it causes this huge buildup of mucus and stuff like that, because you're supposed to. What it does is that it regulates the transfer of chloride back and forth between the mucous membranes. And so when that's gone, when there's this mutation that makes that amino acid go missing, right? There's all this mucus buildup because the chloride can't go back and forth, be regulated to go back and forth, right? And so you get all these, you cough up all this blood, and it's cough, and then you cough up all this blood because all that mucus makes it really a nice, growing ground, right, for bacteria, and then your immune system has to attack the bacteria, and to get there, they break through your lung cells to get to the site where the bacteria is, right? And when that happens, your lungs start bleeding, and it's a very bad situation, all right? So cystic fibrosis is very bad, and a lot of kids don't make it when they have cystic fibrosis. Then there's Down syndrome, also known as trisomy 21. Trisomy 21. There we go. And it involves the 21st chromosome, right? And it's a leading cause of mental retardation, occurring in about 1 in 800 births. The mother's age is strongly influences occurrence. Mental, physical problems, including heart, eye defects, result, and it's the formation of 3 chromosomes at the 22nd chromosome instead of 2. That's why they call it trisomy 21. Then there's familial hypercholesterolemia. Cholesterolemia? Did I say that right? I think so. This one occurs when there's a mutation of the gene on the 19th chromosome, which produces high cholesterol levels that leads to early coronary heart disease in 30 to 40-year-olds. Wow, I didn't even know about that one. Some more genetic diseases. We have muscular dystrophy, also known as MD, right? This happens when you have a mutation in the X chromosome. This muscle destroying disease appears in about age 5 and death by age 20. There's Huntington's disease. Affects the nervous system, leading to the total physical impairment, right? This is a result of a mutation in a gene on chromosome 4, right? That was my dog there shaking, sorry. Just woke up from his deep slumber, I guess. There's sickle cell anemia. We were talking about sickle cell anemia before with the malaria and all that stuff. This happens when the gene on the 11th chromosome has a mutation in it, right? And decreases the oxygen carrying capacity of red blood cells. Hey, but you don't get malaria, right? Very sad. Anyway, there's hemophilia. There was like a Russian prince. Nicholas the Great, was that him? He had a son that had hemophilia. So hemophilia results in one or more defective blood clotting factors that leads to poor coagulation, so excessive bleeding, right? And there's Tay-Sachs disease. And this is a result from the defective hexosaminidase A enzyme, right? Which causes accumulation of gangliosides and leads to mental retardation and loss of motor control and early death. Gangliosides are in the brain, by the way. Like ganglia, right? So those are some genetic diseases. But there's some good things we can use. We can do DNA with, right? Well, for one thing, DNA is it's our genetic information. It's where life is from, right? But DNA can be used in genetic engineering, right? And it permits scientists to cut and recombine DNA fragments to form recombinant DNA, which I was talking about before with the PCR. And then also you can use it to identify a person by examining bands that represent DNA fingerprints. This is actually old. We don't do the examining bands anymore. Now we use STR. Remember, I'm a forensic scientist. We don't use this band thing anymore, right? We use STRs now, which are short tandem repeats. To do DNA profiling, right? And that's what this is right here, right? Which I'll get more into detail about that in a second. So let's first talk about recombinant DNA. So when we're doing recombinant DNA, we have a gene that's in an organism, right? And we're interested in that gene. Really, we're interested in the protein that comes from that gene, or the enzyme that comes from that gene. And the easiest way to study it is to find its DNA sequence in the organism, right? And amplify that sequence so that we can get a whole bunch of that gene, just that gene, not the entire genome of the organism, but just the gene that we care about, right? And then splice it into a plasmid, right? Which is a piece of circular DNA that's in bacteria. But the interesting thing about plasmids is that they're self-replicating and self-expressing, right? And so you can put your gene into the plasmid from the bacteria, and then transform or put that plasmid into the bacteria. And hopefully the bacteria will start making crap tons of the protein or the enzyme that you want to study, right? And so for like 4 or 5 years of my life, this is what I did. And so I did it with two different enzymes. There was the luciferase enzyme that I was talking about back in the previous lecture, where luciferase takes luciferin and makes blue light. And then the other one was MCR, methyl coenzyme reductase, which takes CO2 and makes methane gas, right? Both very interesting for people because one makes light, the other makes a fuel, right, from a greenhouse gas, right? So literally, we would take cells of dinoflagellates for the luciferase, mash them up, right, and extract its genomic DNA, right? So that's the DNA of every gene inside of that dinoflagellate. And then we would then use polymerase chain reaction to amplify out the luciferase gene and then ligate it into a plasmid, like you see here, right? So plasmids come from E. coli. Well, all bacteria have plasmids, but we generally use E. coli plasmids. And we'll get the plasmid out because we want to use that to replicate and express our protein from the DNA that we insert into it. And what we do is that we use these things called restriction enzymes. And restriction enzymes will cut the plasmid in very specific spots, right? Restriction enzymes are named things like EcoR1, BAMH1, those kind of things. And they're part of a thing called the restriction modification system, right? The bacteria have to help fight bacteriophages. So bacteriophages are just viruses that attack bacteria. And what viruses do, or bacteriophages do, is that they will attack a bacteria by inserting its DNA into the bacteria so that it can use the bacteria's machinery, like its polymerases, right, to replicate the viral DNA, right? Because the virus doesn't have that kind of machinery. And so what happens is that in bacteria, specifically like, for example, E. coli, there's this thing called the restriction modification system where bacterial DNA is methylated, right? So we were talking about the structure of DNA, which is that phosphate with the sugar with the nitrogenous base, all connected with the backbone, the phosphodiester backbone. But bacteria have an extra methyl group on its nitrogenous, on its DNA, right? Going up and down the DNA strand, right? And viral DNA doesn't have that, okay? So the bacteria will recognize that the viral DNA is in there and destroy it because the restriction enzyme, right, can recognize the fact that the viral DNA is missing those extra methylations, right, those extra methyl groups, and destroy the viral DNA. Well, we as scientists hijacked that, the restriction modification system in bacteria, and we use it now to cut plasmids in very specific spots, right? And then what we do is we take our, for example, I was talking about the luciferase gene. We take the amplified luciferase gene that we got from the genomic DNA of the dinoflagellates that where the luciferase came from, right? And we'll ligate it into the plasmid where we told the restriction enzyme, right, to cut the plasmid open. Right? So that's what you see here. We use a restriction enzyme to cut a plasmid open. It has sticky ends, right? Because when we cut the restriction, when we use the restriction enzyme to cut the plasmid open, what happens is that it cuts it so that there's overhangs, right? Where the double helix is longer on one side than it is on the other, right? With a certain sequence. And then we'll amplify our gene that we got, luciferase gene that we got from dinoflagellates, and we'll give those sticky ends too, or overhangs too, where one side is longer, of the double helix is longer than the other, right? And it complements the and of the plasmid that got digested, right? With the overhangs. It makes it so that the gene fits perfectly into the plasmid. And once we put the plasmid into the, so this is called insertion, right? This is called insertion. And then we have to have an enzyme that goes in and actually binds it all together. And we call that ligation. Right, ligation. Done with a ligase. Specifically, we use T4 DNA ligase. Man, we're always hijacking viral stuff, aren't we? If only the public knew. But we use T4 DNA ligase, which is from a bacteriophage, a virus, right? And we ligate it all together. Ligating means to bind it all, not bind it together, but make it all one strand again, connected it, splicing it together. And then we transform it back into E. coli. This is called transformation. After we transform it back into E. coli, hopefully, we're successful in that transformation, right? And the plasmid starts to reproduce, making multiple copies of itself. And then, after making multiple copies of itself, starts to go through the transcription and translation process to make the gene that we want, right? Which in this case was the protein we want, which is luciferase, right? My blue light producing enzyme, right? And that's the basis of most recombinant DNA. Using a plasmid from E. coli, using restriction enzymes to cut it open, also using PCR to amplify, make a whole bunch of a gene that we want from a different organism, right? And putting it into the plasmid that we just cut open, then putting that plasmid back into the E. coli, and then hopefully we grow it, right? Make a whole bunch of the cells of the E. coli cells, and then we make a whole bunch of the plasmids, and those plasmids hopefully will replicate themselves and then go through the transcription and translation process to make a whole bunch of more protein, right? So this is called heterologous expression. Heterologous because we're expressing it in a different molecule. Different organism. We're taking a gene, luciferase, from the dinoflagellate organism, and we are expressing it in E. coli, which is a different organism, right? And this is the traditional way of doing recombinant DNA. Now we can do things like use a CRISPR, where we can directly modify DNA, CRISPR-9. But traditionally, this is what recombinant DNA has been. So there are a lot of different drugs that have come from recombinant DNA, right? We have human insulin that's been made from recombinant DNA, which makes sense because remember what insulin was. Insulin was just a polypeptide chain. And we know its sequence, so we can literally just figure out what the sequence is, get its codon, and put it into a plasmid and get E. coli to make a whole bunch of this insulin, and we have insulin now to treat diabetes, right? There's EPO, which is erythropoietin. I have never heard of that before, but it treats anemia. There's HGH, human growth hormone. There's interferon, which treats cancer and viral diseases. All these are drugs that come from recombinant DNA, right? There's tumor necrosis factor, which destroys tumor cells. Antibodies, right? Antibodies transport drugs that are needed to treat cancer. Epinormal growth factor stimulates healing of wounds and burns. Man, where was that at when I needed it for my dog bite? Dog bite right there. I mean, I'm healing on my own. I'm like Wolverine, so I heal up dog bites pretty fast. But I'm even faster if I had that. Epidermal growth factor. Human blood clotting factor treats hemophilia. Interleukin stimulates immune system. Prurokinase, I don't even know what that is, but it destroys blood clots. The influenza vaccine prevents influenza, and then the vaccine, hepatitis vaccine, right? So tons of drugs have come from recombinant DNA. So here's a little bit about the polymerase chain reaction, or PCR, right? PCR. No, that's bad. PCP, no, that's a drug. PCR. There we go. So I talked to you a lot about PCR just a second ago about how I was amplifying the luciferase gene from the genome of dinoflagellates, right? And that's traditionally how PCR was used. Excuse me. But recently you've been hearing a lot about PCR because they use it in COVID testing. At my lab where I work right now, at my lab, they do COVID testing there, right? And they use the PCR test. And literally what it's doing is that it's detecting or amplifying viral DNA, right? And if the COVID-19 virus is present in you, right? And we take a sample from you and do PCR looking for a specific gene, right? That's only in the COVID-19 virus, and it makes copies, and we are able to detect those copies of the virus gene or the viral sequence. Then you could be positive for COVID-19, right? Then there's also the antibody test, which is kind of an immunochemistry test, right? But here, it's polymerase chain reaction. So let's talk about the polymerase chain reaction. So in theory, and PCR is very voodoo, by the way. Someone once told me that, and that person was completely correct about that. PCR is very voodoo. But in theory, PCR can happen with just a single strand of DNA. And then by the end of PCR, you'll have millions of strands of the amplified part of that DNA that you wanted, right? And that's the reason why people use PCR to amplify sequences in very scarce like DNA samples, right? Like if you have like DNA evidence and there's not a lot of it, you use PCR to make a whole bunch of the short tandem repeats I was talking about before, so you can make a DNA profile. That's the strength of PCR. That's what makes it so powerful. But we start out with the strand of DNA that needs to be copied, right? And remember, that's just the part that we want to copy. This strand is actually very long, right? Okay. And that's just the part that we want to copy right there, that center part right there. And recall that when naturally when we do DNA replication, we use an enzyme called helicase. What does helicase do? It unzips the DNA to access the nitrogenous bases inside of the double helix, right? But it's very difficult to control helicase so that you can just unzip exactly where you want it to unzip, right? And when you want it to unzip. So what do we do in recombinant biology or recombinant DNA, right? We actually use heat. And I talked about this before. So in replication, it's helicase, but in PCR, we use heat. All right? We usually bring it up to about 95 degrees Celsius, which is perfect for destroying or disrupting those hydrogen bonds that are holding the double helix together, right? And recall, the reason why we're able to use heat is because we're using a polymerase that is from T. aquaticus, right? That thermophile bacteria, right? That has a polymerase that can survive the heat vents in Yellowstone, right? Those hot heat vents. Boiling water. For some reason it doesn't kill these bacteria. And so since it doesn't kill this bacteria, we know for a fact that the bacteria is resilient to heat. So we were able to isolate this enzyme, the polymerase from T. aquaticus, and use it in PCR. We're not using it yet. It still has to be in the mix, right? But it's there, right? If we were to use human polymerase to do this, wow, it just explode as soon as it got up to like 100 degrees Celsius, right? Or 95 degrees. I mean, body temperature is 37 degrees Celsius, right? So if you just slightly above body temperature, so say 40 degrees, 42 degrees Celsius, your human polymerase would just be destroyed, okay? But Thermus aquaticus can survive 95 degrees Celsius, and its operating temperature, right? It's most active at 7 degrees Celsius, right? Much higher than human polymerase. So after we unzip using heat, right, we get 2 strands of DNA, right? 2 strands of DNA. And then, remember, we still have, we still have the thing like this where it's really long, right? The next step, right, is to actually is to tell the reaction where to begin, right? And this is called a primer. But in replication, same thing happens in replication. It uses an enzyme called primase. Primase tells DNA polymerase, RNA polymerase, where to start, right? But in PCR, we use primers. And what are primers? They're literally very short pieces of DNA, right? Very tiny short pieces of DNA that are single-stranded that match the beginning and the end of the gene that we want to amplify, right? Like so. Right? I don't know which side is the 5' side and which side is the 3' side here, so this is kind of weird. But say that this is the 5' side over here, so a primer will end there, will bind there, and then on this one, which is the 3' side, the up there, which means that side is the 5' side, the primer will bind there, right? This is not where the primer begins, right? So there we go. Once that happens, after the primer happens, right, we bring in, where it's already in the solution, right, we use DNA polymerase. But in natural replication, we just use DNA polymerase. That is terrible. Let me write that again. DNA polymerase. Right? In PCR, we also use DNA polymerase too, but it's a heat-stable DNA polymerase, right? So it's special polymerase. The T aquaticus, we'll just call it TA polymerase, right? And each of these happen at different temperatures, right? Each of these happen at different temperatures. The priming step happens at like anywhere between 50 and 65 degrees, right? My Apple Pencil is just not working today. 65 degrees. And the polymerase part happens at like about 72 degrees, right? But that happens in PCR. Okay, the temperature is always the same in regular replication. So once the primer binds, right, to the sequence that you care about, the polymerase comes over and starts bringing in bases to replicate it, right? And then the process starts all over again, where we heat it back up to 95 degrees. Primers bind again to the so it makes 2 strands, right, again, after the polymerase goes through. And then the DNA is heated again to split it again. And then the primers bind again, right? And then the polymerase comes in and starts replicating, right? Starts replicating. And we get 4 double strands now, right? And it keeps on happening, and keeps on happening. And we do it for about 30 cycles, right? We do it for about 30 cycles. And that's how DNA gets, that's how polymerase chain reaction works, right? So the thing I want you to remember about this is that in regular replication, we start out with a helicase to unzip the DNA. But in PCR, we heat it up to 95 degrees. We're able to do that because we're using C. aquaticus's polymerase, right, which is very heat stable. After that, in replication, we use our cells use primase to tell the polymerase where to begin, but we actually synthesize primers to tell the polymerase where to begin. Those are short pieces of DNA that match the beginning and the end of the gene, right? So we call it the forward and reverse primer, right? And those tell the polymerase where to begin. And then we'll raise the temperature back up again, and normal DNA polymerase for replication, but we use T. aquaticus's DNA polymerase for PCR, right? So remember the natural replication and the PCR, right? And so in one tube, this all happens in one tube, right? One PCR tube, and it's tiny. We're talking like 50 microliter tubes, right? 50 microliter reactions. We have these components. We have water, because you can't have a reaction without water, especially in life. We have primers. Primers, right? Then we have polymerase. Right? Then we have some buffer. The reason why we have buffer in there is because the buffer contains some ATP in it. Right? Then we have DNTP. And what are DNTPs? If you recall from the previous lecture, DNTPs are the nucleotides, the free nucleotides, right? That are not bound to anything yet. So the polymerase pulls those out of the solution and then puts them polymerizes them into the DNA strand. And then last but not least, we have the template DNA. Right? Template DNA. So the template is literally like the genomic DNA from the organism you're trying to get the gene from. Like, for my case, it was luciferase, right? In my case, it was luciferase from dinoflagellates. All this happens in like a 50 microliter reaction. We're talking tiny, tiny volumes. All right, enough about PCR. Let's keep going. So we can use DNA stuff or genetics for genetic testing using PCR. Allows to screen for defective genes, right? For example, breast cancer genes. You can also do DNA fingerprinting with the short tandem repeats I was talking about before, right? You can also, the Human Genome Project for one thing, where 21,000 genes coding for 3 billion bases and 21,000 genes coded for protein, which represents 3% of the total DNA, right? In humans. That was pretty cool. And then, just so you know, much of our DNA regulates genes and serves as recognition sites for proteins, and then has been assigned to function and leading to understanding errors in DNA replication, transcription, and regulation. Right? That is because of that. I think I forgot to say that. So let's do a quick thing about viruses, right? So viruses are small particles that have about 3 to 200 genes. They require a host cell to replicate because they're missing the machinery required to do replication, right? No polymerase is there. right? They contain nucleic acids, right? DNA or RNA, but not both, inside the protein coat, and they do not have any materials such as nucleotides, enzymes to make proteins on their own, right? They have to replicate by invading a host cell and using the machinery and materials for protein synthesis to grow, right? The virus attaches to the host cell and injects its viral DNA, uses the host cell's amino acids to synthesize a viral protein, and then uses the host cell's contents to make viral DNA. Then the cell bursts, and the new virus is released to infect other cells. Man, that's insidious, right? So here you have a virus, and it's attaching itself. Almost looks like the coronavirus. I'm sure that's a coronavirus. And it goes inside the bacteria, the cell here, and it injects its DNA using the DNA, the polymerases and the amino acids inside the host cell to make more DNA. And then the viruses burst, the cell bursts, and the viruses go out and infect more cells. So specifically, let's take a look at viral infection, right? A viral infection begins when a virus enzyme makes a hole in the host cell wall, right there. And then the viral nucleic acids enter and mix with the materials in the host cell. So there it goes, injecting its its DNA into the host cell. And the host cell begins to replicate the viral DNA if it contains... the host cell begins to replicate viral DNA if it contains DNA. But if it's RNA, right, viral DNA produces viral RNA, and then a protease processes the proteins to produce a protein coat to form a viral particle that leaves the cell. The cell synthesizes so many virus particles that it releases new viruses to inject more cells. Right. So I got distracted, but I was saying, if it's just RNA and not DNA that's getting injected in here, right, that gets injected in the cell, then it doesn't need to do any replication. The RNA goes straight into making proteins using the bacteria's polymerases, right? Polymerases. So the other interesting thing too is, remember I was talking about those restriction modification systems. The bacterial genome, right, has a whole bunch of CH3s on it, methyl groups, right, attached to it all over the place. But viral DNA doesn't have that. And so a bacteria can actually destroy the viral DNA using its restriction enzymes, and it knows which is which because of the methyl groups that are on top of, that are attached, to its genome. So here's some viruses that are some diseases that are caused by viruses. And this is actually very interesting because everybody knows about coronavirus now, right? But before you thought that it was like a brand new thing when people were talking about COVID-19 and stuff, but it turns out that the common cold is produced by coronaviruses. There's over 100 types of coronavirus, right? And the one that infected us during the pandemic recently, in the last couple of years, or last year and a half or whatever, right? It's just a brand new strain of coronavirus that we've not seen before, right? And that's known as COVID-19. But coronavirus is a very common type of virus that is readily detected, routinely detected, and routinely studied at public health departments, right? So it's not anything that's super crazy, like Ebola or something. Rhinovirus also causes the cold too. Of course, we have influenza. The virus is the orthomyxil virus. Wow, that's hard to say. Warts, the papovirus, herpes virus, HPV, human papillomavirus, leukemia, cancers, and AIDS. Those are all retroviruses. Hepatitis, hepatitis A virus, hepatitis B virus, hepatitis C virus. Mumps, the paramyxovirus, Epstein-Barr virus, and chickenpox, which is shingles, which is the varicella-zoster virus. Varicella-zoster virus. So I was talking about before how part of the central dogma has in it reverse transcription, right, where you go from protein back to DNA. And how do we do that? We do that using reverse transcriptase, right, which is a viral enzyme. And what happens here is that the virus will inject its viral DNA, right, and use reverse transcriptase to make its viral RNA, sorry, use reverse transcriptase to make viral DNA that becomes a provirus. This provirus goes into a the nucleus of a cell and actually embeds itself into the genome of the bacteria, right, or this cell, and then produces more RNA, right, and proteins. And then those proteins escape, becoming a new virus. All right, becoming a new virus. So a little bit about HIV and AIDS. So all I want you to know about HIV and AIDS is that it is a retrovirus, right? It attacks the T4 lymphocyte cells, which is a part of your immune system. So HIV then is an autoimmune disease, right? It causes an autoimmune disease, which means that it actually attacks your immune system, right? It attacks your immune system. The thing that actually really that people die from when they have AIDS is that they cannot fight off normal infections like pneumonia when their immune system's been compromised by the HIV virus, right? So treatment for AIDS is based on attacking HIV at different points in the life cycle, such as prevention of reverse transcriptase of the viral DNA and protein synthesis, right? And so there's a couple of drugs here. AZT, that is similar to thymidine, and it mimics the structure of nucleosides used for DNA synthesis, which inhibits the reverse transcriptase. And another drug is Lexivia. It's a protease inhibitor that prevents the protein synthesis used by viruses to make more copies, right? So that's what I want you to know about the HIV virus. It is it causes AIDS, and it's a autoimmune disease, right? It attacks your actual immune system itself. Then we have cancer. Over, I have a picture of the Epstein-Barr virus, right? And it causes cancer in humans. Normally, cells in the body undergo orderly and controlled cell division, but when cells begin to grow and multiply out of control, they invade neighboring cells and appear as a tumor. When they invade other tissues and interfere with normal functions of the body, tumors are cancerous, right? So cancer can be caused by chemicals, environmental substances, ultraviolet light, medical radiation, and then also oncogenic viruses, right? These are some carcinogens. Aflatoxin is the liver, aniline dyes, the bladder, arsenic, skin and lungs, asbestos, lungs, respiratory tract, cadmium, prostate and kidney, chromium, lungs, nickel, lungs, sinuses, nitrites, stomach, which is interesting because nitrites are in a lot of things, and then vinyl chloride, which is the liver.